Okay, it's been started. So last class we actually where we was just recapping the all those. Let me come back to last class. Just a minute, okay. So we actually tried to solve to find out the some different point group depending on their or what is the symmetry element present in that molecules. So we discuss about what are CO2 followed by uh, benzene, nickel tetrachloride, nickel 2 tetrachloride complex, and again ammonia, cyclopropane, and some organic molecules like acetone, butene, 2-butene, and also one another interesting was there is the Aline and also bipyridine. We solved those problem and try to find out the sorry try to find out we last class we discussed about those and try to find out the what is the actual point group found in that molecules. Coming to next with the introduction of some molecule complex molecule which is actually geometry will be the octahedral, but they have present some different point group. We discuss about that one cobalt complex with aqueous cobalt chloride complex with cis and trans one, as well as the some where the one chloride and substituted when there is a cobalt two chloride and substituted by the water molecule, water one water molecule again uh, substituted by the chloride, and another was the two water molecule substituted by the chloride, and try to find out the point group in that case. Where we found the one chloride substituted one was the uh, where was which one is the uh, 12 C4V, and other one is that D4H, where the two chloride and substituted at the transposition. And again we initially introduce why the optical activity coming from, what is the chirality. We introduce some little bit about this, the why the chirality is the major factor in the biology. So how we can define in the physical phenomena that molecule is chiral or not. To explain that, we actually introduce some optical activity term. That optical activity comes into the another term is coming, the plane rotation of the plane polarized light. So that plane polarized light, introduction of plane polarized light, which is actually in our normal light, which is actually unpolarized light. Whenever this unpolarized light, that is this one is the we are talking about, where this is the unpolarized light. Whenever it pass through a some crystal, where the one axis actually allow to mm, to transfer the light in particular, and oscillate this light in a one particular direction. As we know that our light is the light has a some wave form. That wave form is actually a sinusoidal kind of wave, so it can oscillate in a particular axis. So let's suppose this is actually the axis where my light direction, and this is the direction of the oscillation of my light. This is something like sinusoidal light. This will be the maxima. This one will be the something like this. it goes on. Whenever it's passed through with a chiral molecules, that chiral molecule have, if there is a one isomers over there, that chiral molecule actually turns this plane polarized light into a another plane. So this is called the um, uh, this is called the optical activity, and then that will be measured by the polarimeter. And depending on the how much my light is actually uh, uh, polarized in certain direction, in a particular degree, we can measure the polarity of the not the polarity, the optical activity, the degree of optical activity. So so our fundamental thing is that the only the plane polarized light that can be uh, deviated from its direction in a certain degree because of the interaction between the chiral molecule. So optical activity found in a chiral molecule. So now coming to the another question. So what is the chiral molecule? Chiral is the term called handedness. So molecules, as we previously discussed, molecules having uh, what is that uh, molecules having super non superimposable mirror image. Sorry about my fumble. Molecules having non superimposable mirror image. Let's suppose we have a one molecule over here. Let's suppose my hand, it has a mirror image like right hand, but it does not superimpose onto that into that molecules into that hand, because of the our two hands are chiral. These two hands one of each other called enantiomers. So the another more interesting thing is that this chiral molecule doesn't hold any symmetry elements except Cn. The molecules of chiral molecules can hold the Cn, but it never exists. It doesn't have any other symmetry element rather than the Cn. I, it doesn't have the I, it doesn't have the sigma, it doesn't have the improper axis of symmetry. Only we can found the Cn. Sometimes we cannot find. Cn means you can go for the identity operator. C1 is the identity operator. You can find somewhere. Everywhere we can find everywhere identity operator. C other rather than the one, n equals to one, we can find some other numbers. But if it doesn't contain any other symmetry element rather than the Cn, then the molecules possibly the chiral molecule. And another thing is that uh, molecules having the chiral center, but we don't find the what is that optical activity. You can find some cases also. Now I'm coming to that point in a first, where there is a one molecule, interesting molecule, which is called hydrogen peroxide. I think professor also uh, discuss about these things. I want to also talk about this. Where is my pen? So hydrogen peroxide H2O2. So what is the structure of the H2O2? It's a kind of open book like structure. Okay. So oxygen over here, oxygen over here. Here is the hydrogen, and here is the hydrogen. It looks like a open book kind of structure. So I have do we have, yes, this is we have. And let's talk about this diary. My hydrogen peroxide looks like, here will be my, sorry, you can show you this is the my hydrogen peroxide kind of structure where where the two two oxygen over here, one oxygen over here, one oxygen over here one oxygen, and another hand is the another oxygen. So now hydrogen is actually over here in direction, and from this oxygen to this direction. So this is a kind of open book kind of structure. Okay, so now if we properly draw it, how can we can draw it very easily? Let's suppose this is my one plane of the book. This is another plane of my book. Okay, here the one oxygen over here, it is connected with the OO bond. Here is the hydrogen, and here is the hydrogen. Okay, so this molecule, let's find out it's the point group. Point group will be point group will be now my process will be the if it is linear or not. Linear, no. Is it cubic? Under cubic point group, obviously it is not. Then is it have any C2? Let's find out. Do we have a C2 over here or CN axis? Is there? Is there any CN axis? Yes, it has one CN axis. How can you find? Here is the axis we are talking about. Or here is the axis we are talking about. Or here is the axis we are talking about. Now I check. Now this is the thing. So this is the axis we are talking about. If we rotate it 120 degree, it's look, this hydrogen goes to this direction, this hydrogen goes to this direction, and it's come back to that direction and it will come over here. So it has C2. Now coming another question is the 2C2 perpendicular to the C2 axis. Basically there is no other C2, so it is no. Now is there any sigma h? No. Is there any 2 sigma v? Basically no. We don't have any sigma. So this is the point group will be. This will be the point group. Point group will be my C2. Okay. Now question is that if my molecule is under the C2 point group, so there should be a chances of optical activity. That's why we actually talk about that. The molecules having only the CN axis, no other symmetry element is present. So that means there is a chances of C2. There is a chances of what is that? There is chances of optical activity, but it doesn't. Opti- it- my hydrogen peroxide molecule is not optically active. So why does it so? So there is a oxygen bond over here which can rotate itself and makes another isomer which is actually enantiomer of this geometry, and that kills the optical activity. and it becomes the racemic mixture. And this bond rotation is very rapid in the room temperature, even in the low temperature, something like minus 10, minus 15 degree. So we cannot find the optical activity in the hydrogen peroxide because of the OO bond rotation. Okay, fine. Now coming to the picture, why the chirality is so important into our system. So chirality actually, for every in biological system we definitely find lots of chirality in coming from the our natural body, in our body or natural any other system, like some protein structure, glucose, all those things, they are chiral in nature. So why this is chiral so important? One question is that, so one answer of that is the molecular recognition answer. One is major answer is the molecular recognition. We have huge number of things in our system, in our nature, but we supposed to take on particular things from the nature which is actually required for our body, not other thing. So for that one only we need some molecular recognition. How the all those microscopic level recognition happens, we are going to discuss it in this session. So first of all, talk about the chirality. So chirality is the molecules having at least one chiral center over there, or and so this chirality actually associated with the enantiomers, and when these enantiomers are equally present, that is called the racemic mixture. Now in our biological system, where we found the one particular enantiomer actually predominantly active in our system, in our body or any natural process. So for that we have to make one particular enantiomers also. So for that we need the molecular recognition process. So to make a enantiomeric access, what is the enantiomeric? How can we make the enantiomeric access? Enantiomeric access. So we have to synthesize the molecule in a chiral environment. Synthesized in chiral environment. Okay, and then so uh, we need to make a chiral environment so that we can actually synthesize one particular enantiomeric access. So what are the natural chiral environment we can find? So we can find some natural chiral environment is the first one is the chiral chiral environment. Chiral environment is that, first of all we have protein. Protein is a chiral molecule. Chiral. We talk about the protein in later part. Second one is the carbohydrate. third one is the nucleic bases. Okay, so coming to the chiral, it protein one. It has a building block is the chiral. Some building block. Building block also chiral amino acids, which is amino acid except one amino acid that is called glycine. Glycine is achiral in nature. Its 3D structure also 3D structure also become chiral. Chiral 3D structure. It is called alpha helix. Carbohydrate, that we know that D-glucose is a is a chiral molecule. The nucleic acid, it is the RNA and RNA. Some RNA has and DNA. They have the helical structure. Helical structure. Okay, so where we find the some helical structure, there should be a chirality associated within it. Okay, so let me. Okay, where we was the nucleic bases. Okay, we talked about the nucleic bases. Those chiral environment actually naturally occur. So using that, we can actually make a chiral environment, and we also synthesize the optically active molecules with the enantiomer excess. Okay, so how we can do that? What is that major factor? Coming to uh, the optical environment we are finding. So how we can recognize that that molecule actually uh, require to synthesize on particular things. So with that, the there's called some enzymes pockets. Enzymes are there. The enzymes is nothing but a metalloprotein. So what is the enzymes? We are talking about the enzymes. So in the environment molecular recognition recognition requires so that the chirality chirality plays crucial role. Okay, so fine. So one example is the enzymatic reaction. Okay, so enzymatic reaction, as you all know, there is called lock and key theory. Lock and sorry, key theory. Lock and key theory. So how it is creates? We have something called enzymes pocket. Let's suppose this one my enzymes. It has a one specific region which is this safe, and we have some over here. This is my enzymes. We have some other. We have this kind of substrate. We have this kind of substrate. We have this kind of. We have another thing is that this kind of substrate, and also we have this this kind of substrate. Okay, so all the substrate doesn't fit with this. This is called lock, and this is the key. So enzyme has a some specific pocket for some specific. This is called substrate. So enzyme is a something like where we find some specific pocket over here. Let's suppose here change the color blue. This region we have something like some molecules present which actually helping to bind the safe of this substrate is only for this one. So this can only binds this substrate and break it down this substrate into a small molecular level, and then that can be absorbed in our intestine, or bigger intestine or smaller intestine. Okay, and this this lock and key mechanism, this molecular recognition over here, actually by this binding pocket, molecular recognition happening in the binding pocket of the enzymes, actually happening by some chemical bonding process. The substrate enzyme interaction, substrate enzyme enzyme interaction happening some particular interaction by bonding, chemical bonding, some particular bonding, that is bonding happening through one particular is that hydrogen bonding, through hydrogen bonding. Second one is that salt bridge interaction, hydrogen bonding. We know all those things. We have some electronegative group. Some, let's suppose this is the X. X can be nitrogen, oxygen, fluoride. Here we have hydrogen, and then we have X dash. X dash can be X or something like this, and then something continuous. This is the hydrogen bonding, and we have salt bridge interaction. Salt bridge interaction. How it's happening? Salt bridge interaction means we have some positive charge ions over here. We have some negative charge ion over here. They can interact to each other and making some ionic kind of bond, which is called the salt bridge interaction. Now, third point is that hydrophobic interaction. Hydrophobic interaction. How this hydrophobic interaction happens? Now, let's suppose I'm writing over here. We have some enzymes over here. Uh, Enzymes. Let's suppose this is my enzymes. This is the enzymes, and this is my red color. This is my substrate over here. Uh, let's suppose you can make it something. We have this is a say. This say are only say, enzyme, and this is a substrate. So this in the biological system, it associated with a huge amount of water molecules. Let's suppose here over here, this is the H2O here, over here. This associated with the huge water molecule. Okay, this is the water molecule, and the enzyme also associated with the huge amount of water molecules. Solvent surrounds its structure. So when they comes together, this is the hydrophobic. These are also hydrophobic. Inner side is the hydrophobic and outer side is the hydrophilic kind of structure. And when they comes together, when it forms together, this is my, and when they binds together, here is the substrate binding through it. This is substrate. This is enzyme. They comes together. The associated water molecule over here, over here, over here. It releases the number of molecules, water molecules. Is entropically favorable. Entropically favorable process. Okay, so and this hydrophobic group can combine together and water molecules surrounds it becomes the entropically favorable process. So because of that, the initially our system was the surrounded by the water molecules, and then this is the hydrophobic substrate and hydrophobic enzymes. It's surrounded by the some water molecules. When those combine together, there is a huge number of water molecules comes out from the system, and it becomes the entropically favorable, and then the hydrophobic part combined together, and then it becomes the it becomes a favorable process, that is called the salt hydrophobic interaction. The another thing is that we have the pi stacking interaction, pi interaction, and 4 will be the pi stacking, pi stacking interaction. 5 will be quick van der Waals interaction, van der Waals speed. VDW. Yes. Okay. These are the interaction we found to make formation of this what is called this word, this this substrate this substrate into these enzymes, and there will be some where we can write it one more times. We have some enzymes over here. This will be my enzymes, and this will be connected to some things. This is my uh, next change my color now. This will be the red, and enzymes will be my red. Enzymes will be the red in color. Where will be this will be the enzymes, and this is the substrate combined together. Oh, so we can write it over here. This is the substrate, and this, let's take another color. Here, this this portion, they have some some interaction, something like something like this is called uh, bonding interaction. Bonding interaction. Another point is that to recognize the molecules, that is the chirality. Chirality. Okay. So how the chirality comes? These enzymes, enzymes, and substrate contains lots of amino acids and carbohydrate. Acid and carbohydrate, which will actually plays a crucial role to actually recognize that, to make a chiral environment, and followed by recognition of the molecule, and forward to make the further reaction. Okay, so now first of all, now coming to the protein. Lots of amino acids is a building block of protein. So proteins, so this is the polymer of amino acids. So amino acids itself is a chiral molecule. This amino acid is a chiral molecule, except except cystic. Okay, except glycine. Sastik Kumar, you are right now the first. Satish, sorry. Satish, are you here? Satish, are you hear me? No. Okay. So, what are the what are the amino acids found in the nature? We have lots of amino acids. Now coming to the list of the amino acids. So acids, those are actually found in nature, which is actually L amino acids. How we can nomenclature this L or D? Now coming to the another rules is called corn rule. CORN, corn rule. So what corn rule says? We have orientation of my amino acid such that my COOH and acid group and followed by R group will be something like that. That the rotation of that in particular COR and N will be the anticlockwise, then it will be the L. So now coming to the in particular way, that is this will be the COOH. This will be my R group over here. This one will be the hydrogen, and this one will be my amine group. And always con rule says rules should be hydrogen should be in back, second will be my CO. This will be the con. This one CO. This one will be the R, and this one will be the N. So this one my 1, 2, and 3 is rotating direction will be this way, and rotation orient this groups orients the groups such that where the it should be CO followed by R's followed by N. N will be the NH2, and this will be my COOH. Okay. So if it is anti-clockwise, then it will be called L. If it is clockwise, it will be called D. D amino acid. So L amino acid. Okay, L amino acid. So now coming to the what are the all amino acids present in the nature? We have some list. Each of the amino acids denoted by its name in a three-letter code as well as the one-letter code. So now coming to the structure of the amino acids. First of all, amino acid structure. Amino acids. One, its basic structure will be something like my COOH, carbon, hydrogen will be the back, my NH2 over here, and this one will be my R. This is the common structure of the L amino acid. Okay, so first of all, if my R equals to, let's suppose R name will be three letter code, one letter code. So the R name, three letter code, one letter code. If R equals to my only hydrogen, that is called glycine. Remember that there will be this R will be the hydrogen, then this center becomes my achiron, glycine. Three letter code is the GLY, and one letter code will be the G. So we have to remember at least one letter code, because of that, to define some peptide we need the amino acid codes. Second thing is that there will be some, if we introduce some CH3 group over here where R equals to CH3, then it is called alanine. Ala, alanine, sorry, alanine. This three letter code is the ALA, and one letter code will be it's A. Now, some uh, if you introduce some other aliphatic group inside our aliphatic chain, that is, we introduce over here CH2, and no, no, not CH2, CH over here, CH3. We are introducing isopropyl group over here. It is called Valley. Three letter code is Val, one letter code is B. Now again we introduce another CH2 group before it, which is actually CH2, CH, CH3, CH3. It's called leucine. Three letter code is Leu, one letter code is the L. Again, there is another isotope, this CH3 group. Let's suppose this CH3 group comes to this at that CH group. So what is will be the structure? CH, CH3, this is CH2, CH3. This is called isoleucine. Isoleucine. Three letter code is the ALI, ILE. One letter code is the I. Okay, fine. Now we shall all introduce the uh, um, aliphatic group over here. Now coming to the aromatic one. First of all, we have to draw the this over here. I'm writing over here. This will be my COOH. Hydrogen will be my back. NH2 will be over here upside, and that will be my R group. Based on the R group, we are actually Nomenclature. The molecule are name, three letter, one letter, one letter. So this will be the aromatic. Now coming to the aromatic. So now if my our group becomes over here CH3 and this is the binding. It's called what is name? Phenylalanine. Oh sorry, structure is the wrong structure. CH2. This is called phenylalanine. Three letter code is the PHE and one letter code is the F. Okay. Now why it is F? Now coming in the later part, it can be denoted as a phi, but P is the proline. Now again we introduce on indole group over here. Then this is the CH2 and this is the called indole group over here. It is called tryptophan. This is called tryptophan. Three letter code is TRP. One letter code is W. Now if you introduce some OH group over, this is my CH2 and this is my bonding site, R bonding site to the C, and if we introduce OH over here, that is called tyrosine. Tyrosine. Three letter code is the TRY. Oh, sorry, TYR. TYR. One letter code is the S. Oh no, Y. One letter code is the Y. S is the serine. Now, if we introduce some polar group, what are the group we can find? Let's suppose in case of polar group, we have some CH2OH that is called serine. Serine. Three letter code is the CR. One letter code is the S. This one is the Y. This is the S. Now we have CH OH over here and CH3 over here. That is called threonine. Threonine. Oh, this will be H over here. So this is name. Three-letter code is the THR. Small letter code is the T. Now another group is that C is 2. This is the CH2, C double one O, and this is the NH2. This is called as, what is that? Asparagine. 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 What is three-letter code? ASN, and one letter code is the N. Now we have another molecule which is over here CH2, CH2, and we have another CH2 over here, and then CO NH2. This is called glutamine. Glutamine. Small letter is the GLN. As the glycine becomes the G, now it is called as E. Okay. So now coming to the next page. So we are talking some other acidic group. This is the acidic group. Acidic amino acids. The R name, three-letter code, one-letter code. Acidic one. First one is the aspartic acid. It is the CH2 COOH. That was the asparagine. Now it is become the aspartic acid. Aspartic acid. This is known as the ASP. Aspartic acid was the ASN. Is the one letter code is the D. HJB. Now what should be another one? It's the glutamic acid. Initially we found the glutamine. So how it structure? We have to introduce another CH2 group and then followed by COOH group. This is called glutamic acid. One letter code is the GLU, and one letter code is that E. Oh, this will be the GLN. This is the, this will be the Q. This is the Q, and then this glutamic acid will be the E. Now some basic functionality also be there. Basic functionality. So basic amino acid. What are the those? One is that we have long chain CH2, 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 and then NH2. That is called lysine. The one three-letter code is the LYS. one letter code is the K. Now if you introduce some guanidine inside over here, once instead of CH2, 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 over here NH2, NH, C double one O, NH2, oh sorry, this is C double one NH. This is called arginine. 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 So one letter code here, ARG, one letter code is the R. We have some other also. Another is the um, imidazole unit over here. One is CH2, where the R is the one imidazole inside this amino acid. This is the NH. This is over again, and then this one will be there. No, something like that. This one will be the orange things. Okay, this is called this name history. Three letter code is the his, it will be the H. Okay, now we have four other amino acid. So other four amino acid. One is that node R. So we'll name three letter code, one letter code. So when R is that one CH2 SH, that is called cysteine. CYS, CYS TEINE. Cysteine, three letter code is CYS, cysteine, and one letter code is the C. Now if you introduce some thioether group over here, CH2, CH2, S, CH3, that is called methionine. Methionine, and three letter code is the mate, one letter code is the M. Now if instead of this S, if we introduce some selenium group over here, that is called selenocysteine. What is that? CH2 ACH, that is called selenocysteine. Three letter code is the CC, says one letter code is the U. Sorry, should be the capital. And we have another one which is called proline. This is the structure of the proline. This one is the structure. This is the carbon over here. Should be my COH. This one my the hydrogen. This is the NH. These are the cyclic one. This is called proline. and the three letter code is the pro, one letter code is the P. Okay, so these are the amino acids which is actually naturally occurring. So all those amino acids, starting from the glycine. Glycine is a very glycine is a achiral molecule. So we have to remind all those amino acids one letter code. So starting from the when the R is equals to hydrogen, it becomes the glycine. CH when the R is the CH3, it becomes the alanine. Three letter code is the Ella, and one letter is the A, is so on and so forth. So these are the aliphatic amino acids. These are the aromatic amino acids where we found some aromatic group attached with attached as a R group. So one is the benzyl group, another is the indole group, another is the phenolic group, which is tyrosine. Now we have some polar group with the aliphatic chain, which is sometimes it is OH and sometimes it is amide, some amide one. One is serine, threonine, asparagine, and glutamine. These are the polar one. Aromatic one is the phenylalanine, tryptophan, and tyrosine. Now we have some acidic amino acids, which is actually aspartic acid, glutamic acid, lysine. Basic amino acids are lysine, arginine, and histidine. Some other neutral amino acid, which is actually named as the cysteine, methane, methionine, and selenocysteine, and followed by the proline. Proline is the cyclic amino acid. This is proline has some unique features. Whenever we are going to discuss about the this proline, proline has a unique unique property. When the amino acids, if you look into the 3D structure, when the amino acid turns, amino acids have some turns. So let's suppose we have some alpha helix, something like this of the amino acids. Now we have some chain where we need to turn over here, and then followed by another helix over here. Where this turns is present, you will definitely find this. There should be proline will be present there. Proline should be there. Proline should be present in tar of the 3D structure of protein with peptide peptide bond. Okay, so this proline can be found always found uh, in the turn of the any some 3D structure of the amino acids. It helps to make turn some direction. Okay, so so what is the role? Although you know these are the amino acids, all are the L amino acids. Now coming the question, what about the D amino acids? So how it should looks like? So D amino acid, we call this when we call it D amino acid. Now called is the D amino acid. When we call it D amino acid, we have molecules something like COOH, carbon, hydrogen backside. In the corn rule, we always supposed to be hydrogen should be in backside. If R is over here, N is 2 is here, then if we call this is the 1, this one will be the 2, this one will be my 3 group. So it is in clockwise direction, so it becomes the D amino acid. Okay, so this is the D amino acid, and if we replace some L amino acid into the D amino acid, there is a certain changes in the peptide bond, and that protein cannot be recognized in our body. Let's suppose we are taking some protein which contains one or two. So it contains may contains the some D amino acid inside it. So it doesn't actually react in our body. It may will not absorbed in our body. Because of that, we have some enzymes which is called peptidase or peptidase. So if peptidase is not recognized in that D amino acids, so it will never break down the enzymes, and then if never break down the amino acid peptide bonds, and that peptide bond if not break down, it cannot be easily digestible in our body, and it will not absorb in our body. So now in our case, uh, we know that in the bacterial cell, where the all those cellular membrane, all those things, these are actually made of my peptides or protein. So if some bacterial cell is there, so there is a peptide bond surrounds over this cell, and that peptide cell will not be easily breakable in our stomach. So that's why we are getting the bacterial infection from the food poisoning, all those things. And how the now question is the how this antibacterial reagent actually works. Actually, antibacterial reagent will be made of some 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 peptidase kind of enzymes, which will actually binds in a one particular amino acids, one particular point, and it break down this amino acid peptide chain. And one particular point, if it is break down, so it is become the easily breaking other peptide bonds somewhere else. And if it is break down, my peptide structure is gone. Now this this bacterial cell can be easily kills by the our body, because we have a lots of HCl over in our body, so it cannot be survive over there. So in the bacterial cell, we have some peptidase as a antibacterial reagent, and then it binds. Peptidase actually binds with the one particular group that is called the arginine. Let's suppose arginine peptidase in a arginine. What is that? Arginine has a some guanidine molecule inside over there. So arginine guanidine molecule over there, some NH N. So what is that? Arginine, Arg, arginine. So we have some R. Yes, this is the NH C double one NH and NH2. So this basic group actually binds with this peptide bond, and there in that peptide bond actually break down over C over this arginine moiety, and then the peptide structure is gone, so it can kill the bacterial cell. Now, so people are also thinking that people are very smart enough. They are actually developed uh, this kind of antibacterial reagent that is called peptidase, containing a antibacterial reagent. Now, if peptidase is not there, uh, if bacteria become over smart than us, they actually mutate this arginine molecule into L-arginine molecule to into D-arginine. Now, if it is D-arginine, so our peptidase actually works in the L-arginine. Right now, it is in D-arginine. Will it be recognized? No, it should not recognized by this uh, by this peptidase, and bacteria become survived over there. So this is basically the mutation of the bacterial cell. So same thing over some, let's suppose some bacteria actually growth. You have the antidotes of that bacteria, but you also infected by that. No, no. Why it is happening? So one thing is that your antibody becomes a weaken. Another thing is that you have the antibody, but that antibody doesn't fit with that bacteria cell. Because of that, that bacterial cell, one particular protein over there, they are actually changed to a defined amino acid, and it is not actually recognizing by our antibody. Okay. Is there anyone right now? No. So this is the thing, how it is happening. In the working of the peptides, we have the some protein chain, peptide chain. Over here, let's suppose uh, this is NH, and this CO. This will be the carbon, this will be the hydrogen. Here is the R group, and here is the other part. For this will be the NH, followed by this. We have these things. This R group. If R group is something, some functional group over here, let's suppose it may be uh, guanidine, guanidine, or serine. We have the OH group. Maybe we have the NH2 group, RNH2, R dash NH2 group, R dash NH group. Then this peptide actually recognize this, that actually binds this. This is the my this peptide is enzyme which will actually binds with this, binds, and this break down this peptide bond, and making the making the peptide bond break, and then it becomes uh, it kills the bacterial cell. If it becomes, let's suppose this is the L amino acid, now it becomes the D amino acids, which is actually mutated by the bacterial cell. So my enzyme will not right now, will not recognized, not recognized, and then it doesn't kill the cell, so bacteria can survive. If D amino acid survived. Okay, so more important thing is that, so this chirality, if chirality is changed, our molecular recognition actually changed. This is the fundamental thing we have to think about it. Okay. Now now some other example is that, this is the one example is the enzyme peptido and peptidase, and this is the antibiotic, another antibiotic reagent, it is the vancomycin. Professor covered this, again I am actually covering once again. for your easily recognition. Second point is the vancomycin. This is the organic molecule. It can this vancomycin has a property which is actually binds with the L-aspartic acid and the L-serine. It's bind with the L-serine and L-aspartic acid and it breaks down the peptide bond. Now, bacterial cell itself mutate to this amino acid, this L amino acid. This is not the 1 year or 2 year process. It is a very long term process. It is mutation happens in a very long term process. So one this Li this all peptides mutate to mutate to mutation happens to D amino acid. It becomes the D aspartic acid, and this will be the my and D serine. Now this vancomycin cannot work over here, so it is not work over here. Vancomycin will not work. That means the tolerant bacterial survival tolerance level of the bacterial cell actually increases. Tolerance level increased increased. So survive in a harsh condition can survive in harsh condition. Okay, so that's how uh, we actually found in the nature where we something we actually developed for the antibacterial reagent. The bacterial cell itself actually developed itself as such that some changing its amino acid L amino acid to D amino acids. The my antibacterial reagent doesn't work over there. Same also think that if bacterial cell is also very smart, then we also become need to be over smart. So we have to right now people are also developing that antibacterial reagent which can also work in a D amino acids. So for that, if that is happened, so bacterial cell also again move to another amino acids which can actually cannot be break down by the whatever the antibacterial reagent we actually providing. Okay, so another point is that we have another point which is number three. Let's talk about color in red. This is number three is that bacterial vibrio colony. Another example is the vibrio colony. Vibrio colony. It has a two phase. One is the growth phase. Second one is the stationary phase. TION phase. Okay, where the my growth will be stopped in the stationary phase. Growth will be zero. Only the metabolism will be there. Metabolism will be there. In that phase, for the in the growth case, we don't have the, so we need, in the growth case we can use the antibacterial reagent. In that case, we have the amino acids synthesis. Further, we can break down this peptide bond, and we actually kill the bacteria. Right now, if we have the some in the stationary phase, bacteria makes itself, this vibrio makes itself in such that we change the L amino acid to D amino acid. In the stationary phase, the product produce the D amino acids, and peptidoglycan cell, the peptidoglycan cell wall made of this kind of D amino acid. In that kind, we are providing some antibacterial reagent. In that case, it's not working over there. Sometimes we have been found, and this bacterial cell becomes the smart enough such that we can know it survived in that antibacterial reagent first conditions. This vibrio colony actually, that's why it survived in a stationary phase a very long term, long time. Okay, why so? In that time there is a no growth of the any bacterial cell. Only they are having the metabolism process, and in that case only D amino acid they are produced, and that cell of the bacterial cell actually made of these D amino acids, which cannot be destroyed by the antibacterial reagent, which is actually made for the L amino acids. Okay, now another example is the another example 4 is the bacteria for the planktons. We can find bacteria, two kind of things. One is the planktons. Those can be recognized, and they form some enzymes, and then they forming some photosynthesis itself, that is called phytoplankton. Phytoplankton. These phytoplanktons also can make their own photosynthesis. Second is that biofilms. These biofilms actually nothing but is a colonization of the bacteria in a certain particular region, and with the help of the protein, polysaccharide, and extracellular DNA and RNA, they actually forming a protecting layer although over the colonies, and that uh, protein layer outside protecting layer actually helps to survive together. These biofilms are actually very survive against the harsh condition because of this extracellular protein over the over over the over the colony, and it also survive against the environmental threats, antibiotic attack. This is good for the bacterial cell, not for the host. Let's suppose this bacterial colonization happen in our body. We are actually providing huge amount of antibody into our body, but it's not working because of that. This bacterial colony actually makes some protein, outer protein layer, protecting layer, which is actually made of some proteins, polysaccharides, antibacterial cell, extracellular DNA and RNA, which is actually initially protect their colony. Whenever we're providing some antibacterial reagent, which is actually very stable in that colonies, and the antibacterial reagent doesn't work over there. So this colonization can happen in human health. Bacterial growth can happen in human health, agricultural system and industrial pipes. Let's suppose water, gas, or oil pipes. There, this kind of bacterial growth happens and colonization happens. We have the some protein layer, and it actually continuously corrode the pipes. Sometimes it becomes showing the leakage. So that is the problem. Now, how we can prevent this bacterial colony growth of the bacterial colonies? The prevention of growth of biofilm. So first of all, one is that introduction of the d-amino acid of d-amino acid. So if you introduce some d-amino acid over here, so we are actually they forming they cannot recognize that d-amino acid to grow the extracellular protecting layer, because of they requires the some protein and polysaccharide and extracellular DNA or RNA. So if it is there, cell will not recognize this d-amino acids direct, and then this peptide will break down. Now, another point is that if d-amino acid is there, which will actually affect the machinery where this peptide protein synthesis happens. Sometimes in our cell we have some protein synthesis happening uh, in our cells. In that case, it affects that, such that this 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 protein synthesis stopped over there, and then it is prevent the formation of the cell. Okay, this is the major thing. Another thing is the metal sensitivity by by this molecular recognition molecular recognition the metal sensitivity. Why we need so that much? Because of in our environment, in, let's suppose in our body cell we need only, let's suppose, iron. for the blood, red blood cell. We need the only iron. We don't need the cobalt. We don't need the copper. All those things. So where in particular case, there's some pocket also be there, which can bind iron as well as it can bind with the cobalt and nickel. But if it is bind with the iron, that the chiral environment of that iron center, or chiral center or ligand environment of that iron center, such that if binds with the iron, it only can go through that cell membrane. There is some doors over there that is called uh, well, it's coming into the in later on. Say the doors over there, only that door, from that door these molecular systems with these metal ions with that particular particular things goes into that door and go inside the cell. No others, no other metals bind with that particular ligand environment, that cannot allow to go through in that particular doors. So and second thing is that concentration. First of all, why we need that kind of ligand environment, all those things and particular things. Okay, so as we know, in our cell there's a very less concentration of metal ions over there. So over there in particular there is a some iron over there. We have to capture it and we have to transfer it in a efficiently into the cell for our making some defined purposes of the metal ions in our body. Same something has been found in some um, siderophore. Siderophore actually cephalopin in bacterial cell. Cephalopin bacterial cell. We have the structure over here. Yes, this is the cephalopin. Some kind. This is called another kind of. Over here, this is the cephalopin molecule, which will actually binds with the zinc, nickel, and cobalt. These three metal ion, if you need this into the cell, this cephalopin, this ligand actually goes out in their channel and binds with the metal, which will actually binds it with this recognition part. This is a this is a kind of, what is that? This is the something like amino acids unit. Okay, this amino acid unit has some specific structure, and these are also specific for this particular individual cephalopin ligand environment. And when it recognized over there, it can goes through with that channel, and then it releases the metal ions inside the cell. Again it come back, again cycle happening repetitively. Now if we change this amino acid into, or this amino acid into the another enantiomers, so if you change that one, so some bacterial cell cannot recognize over here this CNTA, it cannot recognize over that time, and some bacterial case mutate itself such that it can actually recognize this another enantiomers of that amino acids metal complex. And and and, and with that competition, some other bacterial cell cannot do that. If you release this cephalopin with another isomers into the outsider environment, there is a chances of the taking of this metal complex into a by the another bacterial cell. But but they're supposed to be recognized by this CNTA, this holder kind of things. but but it cannot, because of that they are not muted to to take this staphylococcus defined isomers. And the bacterial cell which are actually mutate, they can recognize it, and then it goes through that particular channel and make that metal ions free inside the cell. So they actually increased its what is that increase its survival property. They can easily survive in a harsh condition wherever the low concentration of the metal ions has been found, as well as there is a competition between the surviving of the other bacterial cell. Okay, so today what we actually discuss about, then so same thing also for the nickel. There is a, this is also vancomycin kind of thing. This is another analog where we can actually make the complex with the nickel one, and which actually goes through the channel of that bacterial cell. So what we actually discuss about, we actually discuss about why the chirality and optical activity is a very important. Chirality is very important. So chirality is important because of the molecular recognition in the engine, in the natural process, natural system. So we talk about, for that we talk about the enzymatic process. First of all, this enzymatic process happens some particular lock and key theory, which is actually uh, there's some interaction between substrate and enzymes, depending on that hydrogen bonding salt bridge interaction, hydrophobic interaction, and the pipette taking interaction. There is another important thing is the chirality plays role into that enzymatic reaction. So because of the molecular recognition, so where the chirality comes into the nature, then here comes the nature into chirality is that protein, amino acids, protein and amino acids. Carbohydrate is a very carbohydrate is a chiral molecule, and nucleic acid, that is the RNA and DNA, which is actually 3D structure, which is very which is actually chiral in nature. Depending on the chirality of the nature of that environment, actually recognize some molecule into our natural process. So now coming to the amino acids, we can find some defined kind of 24 iron 24 25 amino acids naturally occurring. All of those are L amino acids, and all of those are optically chiral in molecule except the glycine. Those are the amino acids we talked about. The L and D actually defined by the Kohn rule. Now this is the list of the amino acids. Proline is a very important thing. Is the proline is a amino acid such that which can be found in a turn of the any peptide 3D peptide chain, 3D peptide chain. What turn is observed, there proline will be found. The proline actually helps for the turn of the peptide bonds. Now, if for the molecular recognition, if you change the some molecules, let's suppose we have the some amino acid is L amino acids. For bacterial cell growth, this happens in a L amino acid environment. If you provide the D amino acid, bacterial cell cannot grow over there. And sometimes if you bacterial growth stopped by the peptidase or something antibacterial reagent, where the some bacteria can survive over there, because of that bacterial cell actually made of some defined amino acid which will actually not work under this antibacterial reagent. So this is happens because of the mutation of the bacterial cell. So these are the some examples: vancomycin and the peptidase enzymes, vancomycins, vibrio colony, all those vibrio colony, survival of the vibrio colony because of the in stationary phase they make some D amino acid to form the its peptide cell membrane. and it's prevent the degradation of the peptide bonds. So and then it becomes survive into that in the harsh condition. In a biofilm, in case of the bacterial cell where the phytoplankton or biofilms actually grow, they are actually colonize make the colony, and in over that colony they actually form some peptide polysaccharides and RNA protecting layer, DNA RNA protecting layer, extracellular DNA RNA protecting layer, which will actually prevent to attack by the antibacterial reagent, and which can be stabilized by the breakdown by the providing some defined amino acid into the making the colonization. If it will make the stopping the um, amino acid synthesis into by their cell by providing some d amino acids which can react with the masonry of the where the protein has been forming. So in that case we can stop the growth of the biofilms. And another major factor is the metal ion recognition into the biological systems. For that we actually introduce some over here is the cephalopin cephalopin or cephalopin molecules, which actually helps to bind the zinc, nickel, and cobalt ion into the and bring it into the bacterial cell. And to there will be some competition between some other bacterial cell. In that, some bacteria actually make their staphylococcus enzyme something which can actually uh, in a defined amino acids, then traditional L amino acids, and then these D amino acids actually recognized by their CNTA. This is recognition pockets that is also actually changed to move to a different direction, so that they can actually take those molecular those metal complex with the ligands, so it can go through into the cell and uh, use these metal ions into the cell. They can release the cell, and uh, the concentration of the metal ions into the cell actually maintained over there. Okay, so to that I will stop over here. Let me stop the